The collagen is the most abundant protein in the human body. Is extensively modified by post-translational modification. So first, most abundant protein in the human body. Then it's modified by post-translational modification. So after the translation, it does undergo modification. Organizes and strengthens extracellular matrix. Okay, organizes and strengthens. Okay, so support and organization of the extracellular matrix. It's from type 1 to type 4 are most common types of in the humans. From type 1 to type 4. So 1, 2, 3, 4. Type 1 is found in the skeleton. Type 2 in the cartilage. Type 1 skeleton. Okay, basic. Type 2 cartilage. Okay, close to it, cartilage. And then type 3 is the arteries. Type 4 is the basement membrane. Okay, these two are kind of also related. Like the arteries has some kind of basement membrane. Yeah. So anyway, type 3 is arteries, type 4 is type 4 is found in basement membranes. For type 1, it's most common. It's because it's the skeleton. Yeah, it makes sense. I mean, it's most common, like 90%. It's in the bone, and we have a lot of bones made by osteoblasts. It's made by osteoblasts, makes sense. And also it's found in the skin, tendon, dentin, fascia, cornea, light wound repair. Light wound repair. So it's found in the bone, in the skin, in the tendon, in the tendons, in the dentin, fascia, cornea, light wound repair. It's type 1 bone and tendons, makes sense. It reduces the production in osteogenesis imperfecta type 1. So there's a problem with its production. Like there's a reduced protection of type 1 collagen that happens or that occurs in the osteogenesis imperfecta type 1. So if a patient has an osteogenesis imperfecta type 1, then he or she could have decreased production of type 1 collagen. Okay? Okay. Let's see. Its type 1 collagen secretion in the first stage of matrix formation is mediated by osteoblast, as we said. So all the credit goes to osteoblasts. A 57 years old man receives this sintered titanium oxide implanted during total hip arthroplasty following traumatic injury. During the first 6 weeks following surgery, bone ingrowth anneals to the bone metal interface in both the femur and the acetabulum. Okay, during the first 6 weeks following the surgery, the bone ingrowth, okay, so the new growth of bone, anneals to the bone metal interface in the bone of the femur and acetabulum, thus securing the prosthesis in position. So, like a new bone formation around the metal prosthesis, okay, the bones are growing all over that. So, it says that type 1 collagen, to maintain the prosthesis in its place. Wow. Type 1 collagen secretion is the first stage of matrix formation is mediated by which of the following cells? So which cell is going to produce the type 1 collagen at such a pace? That's the osteoblasts. For type 2, that's second, it's found in cartilage, including hyaline cartilage, or vitreous body, or nucleus pulposus. So hyaline, vitreous body, nucleus pulposus, that's type 2 cartilage. Okay. Type 3, it's in the reticulum. Reticulum, that's um, in the skin, in the blood vessels, in the uterus, in the fetal tissue, in the early wound repair. So not late, early wound repair. If it's late wound repair, then it's type 1. If it's early wound repair, then it's type 3, okay? So in type 3, it's deficient in vesicular type of Ehlers-Danlos syndrome. So Ehlers-Danlos syndrome could have a vesicular type, okay? And within the vessels, we should have had type 3 collagen, and that's normal, but it is deficient in Ehlers-Danlos syndrome, that's type 3 collagen. Type 4 is the basement membrane basal lamina, basement membrane basal lamina, that's the glomerulus, cochlea, and lens. So it's found in the basement membrane or in the basement basal lamina, and that's in the cochlea or glomerulus or the lens, okay? So type 4 under the floor, that's basement membrane. Okay, defective in Alport syndrome, that's related to the kidney, okay? That's related to the glomerulus all over here. So is targeted by autoantibodies in good posture syndrome. That's second. So two of these are related to the glomerulus. the Alport syndrome, we do have a defective type 4 collagen. And in Goodpasture syndrome, we do have also antibodies going against the type 4 collagen. Okay, so Goodpasture syndrome, auto antibodies against the type 4 um, collagen. Okay, the myofibroblasts are responsible for secretion. What is the thing that secretes type 4 collagen? That's the myofibroblasts. Myofibroblasts. They're responsible for secretion, and that is also called the proliferative stage. The secretion stage is the proliferative stage, and also they are responsible for the wound contraction because they do produce type 4. Okay, so myofibroblasts are responsible for secretion of the type 4 collagen. That's the proliferative stage, and also wound contraction. Okay. Now let's see. For type 3, the one that we talked about was supposed to be in blood vessels, uterus, fetal tissue, and early wound repair. It's also found in spleen and lymph nodes. Also found in spleen and lymph nodes. Okay. Now we're talking about the collagen synthesis and structure. So first we have the synthesis, then hydroxylation, then glycosylation, then exocytosis, then proteolytic processing, then assembly, alignment, finally cross-linking. So for the translation of collagen alpha chains, that's pre-pro collagen, usually glycine XY or GLYXY. X is often proline or lysine, and Y is often hydroxyproline or hydroxylysine. Collagen is one third lysine. Glycine content of collagen is less variable than that of lysine and proline. Let's see, what does it say over here? So many of the amino acids designed XY are proline residues essential for alpha and helix formation and string structure. Introduces the bend into the polypeptide chain. Nice. Okay. Lysine. Other X and Y amino acids from clusters of hydrophobic and charged regions on the surface molecules that enables cross-linking um, to other collagen molecules. Lysine is commonly used to collagen. Okay. Okay, so it seems like the very first step of collagen synthesis is the translation of collagen alpha chains, okay? The collagen alpha chains has another name. First, it has collagen alpha chain name, and then it has pre-pro collagen, pre-pro collagen. It's usually the glycine XY, okay? X is often proline or lysine. So this X could be one of two, could be proline or lysine, okay? While the Y, like this Y, is supposed to be hydroxyproline or hydroxylysine, okay? And in like collectively they're called like lysine XY, okay? Collagen is like third of it, third of the collagen is glycine, okay? So the glycine content of collagen is less variable than lysine and proline because as we said, that this is like the structure of the pre-pro collagen. So it has lysine, it's third of it, of the collagen is glycine, and it's less variable. It's kind of fixed that we do have glycine, so it's less variable. But the X and Y, the X, says it's either proline or lysine, and the Y is hydroxyproline or hydroxylysine. So basically, we do have a lot of options. That's why they're considered more variable, okay? And that's type 1. That's step 1, I mean, pro alpha chain backbone. So pro collagen. It has glycine XY, and the X could be two things, and the Y could be two things, and we talked about those. Yeah, let's see. We have the glycine content, I mean, we already talked about that. So we have the hydroxylation. The hydroxylation, that's of a specific proline. So specific proline, that's a component of the X, okay? That's one of the options for the X. Uh, so hydroxylation, specific proline, and lysine. Okay, that's the other option for the X. So it's the hydroxylation of a specific proline or lysine residues, and lysine residues requires vitamin C. Okay, hydroxylation, just for memorization, requires vitamin C. So if we do have a deficiency, then it's scurvy. If we do have a deficiency in that, then it's scurvy. That's type 2. There you go. Step 2, that's the hydroxylation of proline and lysine, and it requires vitamin C. So um, proline and lysine, they are on the X side of the pre-pro collagen. Okay, so that's for hydroxylation. For glycosylation, glycosylation of pro-alpha chain hydroxylysine residues and formation of pro-collagen, not like pre-pro. No, the pre goes out. It's only pro-collagen via hydrogen and disulfide bond. Triple of helix of 3 collagen alpha chains. Triple helix of 3 collagen alpha chains are problems forming triple helix that leads to osteogenesis imperfecta. So let's see the glycosylation. First, because it is called a glycosylation, so it basically contains some sort of sugar, okay? And some end product of it is the triple helix formation. So triple helix like 3. This is 1, this is 2, and this is 3. So all of these 3 um, lines are coiled together to form the procollagen, okay? And the procollagen, if we read again, it's the glycosylation of pro-alpha chain hydroxylysine. This is what we said. And formation of procollagen. How do we do that? How do we like make this end product? That's by creating a new bonds. These new bonds are the triple helix of 3 of okay. These are the hydrogen bonds and disulfide bonds. So two kind of new bonds are forming. They are the hydrogen and disulfide bonds. They are the ones that are creating the triple helix. So we're bonding these triple lines together. Okay. Problems forming triple helix. So if we do have a problem with this end product, with this triple helix, then it is osteogenesis imperfecta. If we do have a problem with, the procollagen synthesis, if we do have a problem with creating this triple helix formation, either it's the glycosylation or like the creation of procollagen by the hydrogen disulfide bonds. Okay. Number 4, we do have number 4. It's over here. That's the exercise doses. So number 4 is the exocytosis. Okay, exocytosis or procollagen into extracellular space. Exocytosis of procollagen into extracellular space, and it makes sense because it occurs over here. Basically, the exocytosis is just like letting the procollagen outside of the cell. So this is the cell membrane over here, and exocytosis over here, and we are moving out the pro collagen. That's the triple helix. That's the triple helix. Okay, so yeah, that's exocytosis. Now we have proteolytic processing. Proteolytic processing. It's a cleavage of disulfide-rich terminal regions of procollagen. So as we said, this was supposed to be called the procollagen. Okay, what we do is that we cleave those two side uh, parts because they are rich in disulfide. Okay, so we do the cleavage of disulfide rich terminal regions of procollagen leads to insoluble tropocollagen. Okay, this would lead to tropocollagen. That's the only central part remaining. It's insoluble and it's called tropocollagen. How did we do that? By the cleavage of procollagen C and N terminals. Then 6, assembly and alignment. The collagen assembles in fibrils and aligns in cross-linking. So it assembles, like gets together in fibrils, in the shape of fibrils, okay? And aligns for cross-linking, like gets itself ready. Finally, we have the cross-linking. Okay, so 6, self-assembly in collagen fibrils. Self-assembly in collagen fibrils. The For the cross-linking, it's reinforcement of staggered tropocollagen molecules by covalent lysine and hydroxylysine, that's cross-linkage, by copper-containing lysyl oxidase. Cross-linking reinforcements of staggered tropocollagen molecules by cobalt. Okay, so we do a reinforcement. This is a process of strengthening. That's the cross-linking, strengthening of reinforcements of what? Of the staggered protocollagen molecules by covalent lysine, hydroxylysine cross-linkage. So we do this cross-linkage using the covalent lysine hydroxylysine by copper containing lysyl oxidase. So there's an enzyme called copper containing lysyl oxidase. Okay, copper containing lysyl oxidase. It works by reinforcing the staggered tropocollagen by creating a covalent lysine hydroxylysine cross linkage. To make the collagen fibers, that's end result, to make the collagen fibers, cross-linking of collagen increase with age. Like this process of cross-linking, it increases with age. And problems with cross-linking, just like Menkes disease. If we do have a problem with the last step, that is um, the covalent lysine hydroxylysine cross-linking, then it's the Menkes disease. It's a problem with the collagen. Number seven, formation of cross links stabilized by lysyl oxidase. Okay, this is the enzyme stabilized by lysyl oxidase, formation of cross links. And there we go, cross links. This is a cross link, and this is a cross link, and this is a cross link, and this is a cross link. And all over the place, now we have a collagen fibers because it was previously ready for assembly and uh, getting into the final shape. So just this was basically a fibroblast. We know that the fibroblasts are the ones like rich in creating and synthesis of collagen. Okay, we have the nucleus, nothing in the nucleus other than just the DNA and you know going out as an mRNA. That's basically for the collagen and going into the rough endoplasmic reticulum, ending up yeah, and within the rough endoplasmic reticulum, all of this occurs, and then it's moved to Golgi, then it's moved as exocytosed particles, and there we go, we're done. The primary structure is peptide bond between amino acids. Of course, yeah, peptide bonds between amino acids because we said that the amino acid that we have is proline or lysine and hydroxyproline or hydroxylysine. Okay, secondary structure is the hydrogen bond. That's the alpha helix has hydrogen bond after every fourth AA. B sheet has hydrogen bond between every residue of antiparallel strand. Okay, so it makes sense. Over here, the proline and lysine were the basic amino acids. So that's the primary structure because we have um, um, peptide bonds within. But if we have hydroxyproline and hydroxylysine, that's the hydrogen bond between those. And it's an alpha helix. Um, bond after every fourth AA. Beta sheet has hydrogen bond between every residue of antiparallel strands. We have the tertiary structure is the overall shape that a single polypeptide chain assume after compact folding of secondary structures. Tertiary structure maintain many forces by including ionic bond, hydrophobic interaction, hydrogen bond, disulfide bond, um, disulfide bond is a very strong covalent bond between two cysteine residues within the same polypeptide chain that enhances protein ability to withstand the denaturation. So basically, if it's a primary structure, then it only has peptide bonds. If it's a secondary structure, like within the development, it has like now, well, a great thing, it has hydrogen bonds. But if it's a tertiary structure, then of course it's going to have so many bonds, just like ionic bond, hydrophobic bond, hydrogen bond, disulfide bond. And the most strongest one is like the disulfide bond because it occurs between two cysteine residues and they enhance the protein's ability to withstand denaturation so the protein doesn't denature easily anymore. Okay, so pro before this we have pre-pro alpha chain which has amino acid signal sequence at N terminus facilitates in ribosomal binding to the endoplasmic reticulum. Okay, so whenever it's a very start, then we have pre-pro, pre-pro. That's the basic. So the hydroxylation helps to attain maximum tensile strength. Disulfide bond is an interchain disulfide bond formed at the N and C terminal globular region, helping stabilize them. Special interaction disulfide bond also formed at C-terminus between three pro-alpha chains, aligning the helical domain into the conformation. Favorable triple helix formation, the same thing. Disulfide rich is an N-C-terminal polypeptide globular region, non-helical region, removal by pro-collagen peptidase. We have the covalent bond. It's oxidatively deaminates lysine and hydroxylene residues, forming the reactive aldehydes that create covalent cross-linking. Catalyzes the conversion of amino terminus of lysine residues to an aldehyde in collagen fibers. Extracellular collagen fibers are strengthened by this mechanism. Tropocollagen self-assemble into collagen fibrils. Tropocollagen, okay, that's the final product, right? Mm-hmm. Yep. Triple collagen is the final product. Self-assemble into collagen fibrils, which subsequently cross-linked by lysyl oxidase, that's the enzyme, into mature collagen fibers. And that should be it. Mm-hmm. Did we talk about this one? Yes, we did.